This is lecture 5, covering chapter 3, the molecules of cells. This week, we are going to be looking at organic compounds, those compounds that are made mostly of carbon. And we're specifically going to be looking at 4 types of organic compounds: carbohydrates, lipids, proteins, and we're going to touch briefly on nucleic acids, which we'll cover in greater depth in chapters to come. So let's start with an introduction to organic compounds. Organic compounds are those compounds that are made primarily of carbon. If you're a sci-fi fan, you've probably heard the aliens refer to us as carbon-based life forms. This is basically what we're talking about. The word organic refers to living compounds, and we're going to be talking a lot about carbon because that is mostly what organic compounds are made of. So life's molecular diversity is based on the properties of carbon. So let's take a little closer look at carbon. Now, almost all the molecules that a cell makes are composed of carbon bonded to other carbons, as well as the atoms of other elements. Carbon-based molecules are called organic compounds. And take a look at carbon. So carbon has an atomic number of 6. So there are 6 protons in the nucleus of carbon. And there are generally 6 electrons in the electron shells. Notice there are 2 electron shells. The first one is full. The second electron shell, that valence shell of carbon, has 4 negatively charged electrons. This makes carbon a great atom to bond to because there are 4 open electrons. So carbon can form 4 different bonds. Now think back to what we learned last week when we were talking about all of the different elements in chemical bonding, and try to figure out what the easiest thing for carbon to bond with would be. We learned about the special element. It's kind of just a little proton floating around with a little electron. Hydrogen, right? Hydrogen wants to bond once. It has 1 electron. That first valence shell, or that first shell, can hold 2 electrons. So hydrogen is pretty easy to bond to. So one of the most simple compounds that carbon can make is methane, which you'll see in the bottom right-hand corner. The chemical formula for methane is CH4. C stands for carbon, and then H with the subscript of 4 means that that carbon is bonded to 4 hydrogens, and that's what you see on your screen. So one of the easiest compounds, one of the easiest organic compounds to make is methane, carbon bonded to 4 hydrogens. And that's what you see here. So methane and other compounds that are composed of only carbon and hydrogen are called hydrocarbons. They're so prevalent, they have their own name, hydrocarbons. Now carbon, with those attached hydrogens, can form chains of various lengths. So carbon can actually bond to another carbon, which can bond to another carbon, which could bond to another one and another one if it wants. Sometimes they make short chains, sometimes they make long chains, and then any extra electrons that aren't bonded to another carbon will bond with hydrogen. And so that's a simple carbon chain, just a chain of carbons with the empty or the unbonded electrons then bonding to hydrogen. And they can form various lengths. They can also form various shapes, so they don't always have to go in a straight line. You'll see some compounds like butane do, where carbon just keeps bonding to carbon in a straight line, but some, like isobutane, will branch off. So we can have different shapes, we can have branching. We can also have both single and double bonds. So if you remember from last week, a single bond is where each atom shares 1 electron, resulting in an electron pair, so 2 electrons that are being shared between those 2 atoms. That's a single bond, represented here by a single line. They could also form double bonds, where each atom is sharing 2 electrons. So 2 electrons from one, 2 electrons from the other, for 4 total. So 2 pairs of electrons, 4 electrons being shared. That's a double bond, represented by the 2 lines here. So you'll see also that double bond can move. So these are almost essentially the same thing. If you take a look, the chemical formula for 1-butene and 2-butene would actually be the same. The chemical formula, since there's 4 carbons, would be C4, and then there's 8 hydrogens, so H8, so C4H8. Count them in both 1-butene and 2-butene. The chemical formula would be the same, but notice that structurally they're a little bit different. This is going to be important, all right? But for right now, keep in mind that double bond can move, and we can have both single bonds and double bonds. They can also form rings. You'll actually see a lot of these carbon rings in nature. And also notice in this picture here that there's 2 different ways to draw this. So we have the more detailed version on the left where you actually see all the carbons and all the hydrogens that they're bonded to. Now take a look just to the right of that. You'll notice that there's kind of this empty ring. We can make some assumptions about this. So when you see a ring like this, at each of the corners, you are meant to imply that that is a carbon. So wherever you see a point, that's a carbon. And if there's nothing written around it, assume that any extra electrons are bonded to hydrogen. Now take a look at benzene. You'll notice it's very, very similar. Again, wherever you see a point, assume that that is a carbon. Anything else that's not written, assume it's bonded to a hydrogen. The difference here is you'll notice the extra line. That extra line means it's a double bond. So they're saying the same thing, just a different way of looking at it. All right, now if you think back to this, I mentioned the chemical formula of both 1-butene and 2-butene would be the same, C4H8, right? So if you're just looking at the chemical formula, you might not know that there's a difference. But if you look at the carbon skeletons here, you'll notice the double bond is in a different place. This is what we refer to as an isomer, compounds with the same chemical formula, but different structural arrangements. And believe it or not, this actually, well, believe it because it's true, these structural differences cause a difference in how they function. And we're going to notice that a lot throughout the entire semester, that structure plays an important role in function. So there are 3 types of isomers that I would like you to remember and understand. The first one is a structural isomer. So these are ones, again, same chemical formula, but the shape is completely different from one another. So if you take a look at the 2 examples here for structural isomers, you'll notice that each has the same number of black atoms and the same number of gray atoms, but the way that they're bonded is completely different. Those are structural isomers. Now take a look at the next level, geometric isomers. Again, same number of black atoms, same number of gray atoms, same number of red atoms. But in these ones, notice that 1 red and 1 gray are switched. Those are geometric isomers. So essentially they're kind of the same shape, but 2 of the atoms are switched. So now instead of having a red and a gray on top and a red and a gray on bottom, now you have both reds on top, both grays on the bottom. Those are geometric isomers. Now, if you look at the last one, enantiomers, these are basically mirror images of each other. And you know, it kind of seems like you could just flip one over and it wouldn't really be any different, but it actually is. It actually does play a difference. So enantiomers mirror images of each other. So those are your 3 types of isomers. Now, there are a few of these groups, of these chemical groups, that are very important to know. There are a few chemical groups that show up quite a bit. So a few of these organic compounds that we're going to see a lot in organic beings that are going to play a very important role, because each of those chemical groups, because of their chemical formula and because of their structure, are going to affect the function, and they're going to affect the function in a reliable way. So let's take a look at these. Now, the unique properties of an organic compound, of a living compound that's made up of mostly of carbon, are based on the size and shape of the carbon skeleton and which atoms are attached to that carbon skeleton. I said that in its most simple form, organic compounds are rows of carbon with attached hydrogen, but not just hydrogen bonds to these carbon skeletons. It can be other atoms. And depending on which atoms they are and where they're bonded and how long that carbon skeleton is going to determine the properties of that organic compound. And there are 6 chemical groups that are going to play a very important role in determining the properties of organic compounds. So we're going to take a look at these 6. But take a look first at an example here. So the difference between testosterone and estradiol. Testosterone is known as a male hormone, and it typically will cause the animal to show male traits, where estradiol will cause female traits. Notice that the structure is fairly similar. Testosterone and estradiol really aren't that different. There's just a few little differences. You know, take a look down at the bottom here, where testosterone has an oxygen double bonded, where estradiol has an OH group that's single bonded. Testosterone has that CH3. Just minor differences, but it causes a big difference when we're talking about the properties of the organisms that it's affecting. All right, so like I said, there are 6 of these chemical groups that are important for you to know. The first 5 are called functional groups. The 6th is the methyl group. And the first 5 are grouped together and called functional groups because they share common properties, which we're going to talk about. And then the methyl group is also very important, but it's a little bit different than the first 5. So let's take a look at the first 5, our functional groups. Now, my tip to you is you're going to keep seeing these for a couple of weeks. So it's helpful to memorize them, or at least to have a really good cheat sheet handy to use, because we're going to be referring to them fairly quickly, and it helps if you know what the functional group is called, what its chemical formula is, what its shape is, and what it does. Now, functional groups affect a molecule's function in a characteristic way. So if we have that OH group, we know it's going to be affecting the molecule in this particular way. If we have this other group, we know it's going to be affecting the molecule in the same way. So let's take a look. Here are our functional groups. We have our hydroxyl group at the top, which is a carbon skeleton that is bonded to the functional group is the OH group. So OH single bonded to a carbon skeleton. That's our hydroxyl group. The carbonyl group is a carbon double bonded to an oxygen. Carboxyl group is COOH, so carbon, 2 oxygens, and a hydrogen bonded to the carbon skeleton. We have our amino group, which is NH2, nitrogen and 2 hydrogens bonded to our carbon skeleton. And the phosphate group, which is an oxygen, phosphorus, and 3 oxygens with a negative charge that are bonded to our carbon skeleton. Notice that these functional groups can bond in different ways, but that chemical group, what it's made up of, is going to stay the same. Now here's some homework for you. I mentioned that each of these chemical groups affects the molecule in an expected way. So I want you to print this out, write it down, write down what the chemical group is, and then make an extra column for how it affects the molecule. Look it up in your book and fill that in. So figure out, if we have a hydroxyl group, if we have an OH bonded to our carbon skeleton, what does that do to the molecule? If we have our carbonyl group, our C double bonded to oxygen that's bonded to our carbon skeleton, what does that do to the molecule? So there's your challenge. I want you to write it down, look it up in your book, and figure out how each of these affects the molecule. And then keep that sheet handy. It's helpful if you memorize it, because we are going to be referring to these quickly in future lectures. Now, I mentioned that these first 5 groups, referred to as the functional groups, are grouped together into those 5 because they share some similarities. One of those similarities is that all of our 5 functional groups are polar. If you remember from last week, We talked about how those bigger atoms are stronger, so they can pull electrons closer to them, which means the electrons, which are negatively charged, are not equally distributed throughout the compound. So think back to water. Water was a polar compound, where oxygen is so much bigger and stronger than hydrogen that it can pull the electrons closer to it. So if you're looking at a water molecule, the side where the oxygen is pulls the negatively charged electrons closer, so the side with the oxygen is slightly negative, where the side with the hydrogens is slightly positive. That was a polar molecule, where one side has a slight negative charge, one side has a slight positive charge. That's a polar molecule. Our functional groups, take a look here, they're polar. So one side of the functional group will have a slight negative charge, one will have a slight positive charge. This actually makes them what we call hydrophilic, which is water loving. That is compared to hydrophobic, which is water hating. And hydrophilic, those water loving molecules, like our functional groups, are soluble in water. That means that they can break down in water. So that is why we group these first 5 together, because they are polar and they are hydrophilic. Our 6th group is called the methyl group. This is just as important as our 5 functional groups, but it's actually kept separate because it is non-polar and it's non-reactive. Now, it's non-reactive, which means that it's not directly affecting the function of the molecule like our first 5 functional groups are. However, this methyl group is still going to be bonding to a carbon skeleton, affecting its shape. And remember that shape affects function. So when the methyl group bonds to a carbon skeleton and it changes the shape of that skeleton, it is indirectly affecting the function. So our methyl group does still affect the function of a molecule. So there are your 6 chemical groups that are very important to life. You're going to be seeing these 6 groups quite a bit. So again, make a little cheat sheet. Make sure you understand if I say methyl group, if I say hydroxyl group, what that means and what it does to the molecule. Now, cells can make large molecules from a limited set of small molecules. And when we put all these small molecules together and we make these large molecules, we get 4 primary classes of molecules that are important to organisms. And we're going to briefly discuss each of these 4 types of organic molecules. So we're going to talk about carbohydrates, lipids, proteins, and again, we're going to briefly touch on nucleic acids today. Nucleic acids are so important, though, that we're going to spend a lot of time on them in later chapters. So the 4 classes of biological molecules contain very large molecules. We refer to these as macromolecules. The prefix macro means big. Compare that to micro. Micro means small. When we say macromolecules, we just mean molecules that are very big. And our organic molecules here are macromolecules. They are very big. They're also polymers. If you think back to previous lectures, I mentioned, think of a paperclip. One paperclip is a monomer, so that is one piece. If you start linking those paperclips together in a chain, that whole thing is referred to as a polymer. So monomer is one piece, polymer is a whole chain. So these biological molecules that we're talking about today are polymers. So they're going to be a bunch of different molecules that are strung together. Monomers, those individual pieces, each individual paperclip, are linked together to form polymers through what's called dehydration reactions. Now think about that word dehydration. If you're dehydrated, that means you don't have enough water. That's basically what a dehydration reaction is. In a dehydration reaction, we're going to be removing a water molecule and linking things together. Polymers are broken apart by a process called hydrolysis, which is adding water. So if we have two monomers and we want to link them together, we're going to do a dehydration reaction. So we're going to remove a water, which then links them together. If we want to break a polymer apart, that is through a process called hydrolysis. So dehydration reactions link them together. Hydrolysis breaks them apart. We're going to take a look at a picture of this in just a minute. These reactions are mediated by enzymes. These dehydration reactions and hydrolysis require a lot of energy. Enzymes help to speed up those chemical reactions. For right now, that's all you need to remember. We're going to talk about enzymes in depth later on. So let's take a look at a picture. So here we have a short polymer and we have an unlinked monomer that we want to join together. Notice that we have an OH at the end of the short polymer and an H at the end of the unlinked monomer. OH and an H together could make H2O, or water. So here's where our dehydration reaction is going to come in. We are going to remove the H and the OH. We're going to make a water molecule. So we're going to take that water molecule out, and then the end of the short polymer and the unlinked monomer are going to join together to make a longer polymer. That's a dehydration reaction. Now we're going to take that longer polymer and we want to break it apart. Again, this is hydrolysis, where we're adding water back in. So we're basically doing the opposite of what we just did. We're going to add water in, so we're going to add an OH group to the end of the short polymer. We're going to add a hydrogen back to the monomer. It's going to break it apart, and it's going to actually then form the shorter polymer and the monomer. So again, that's hydrolysis to break a bond. All right, so that's the background behind these four organic groups. So let's briefly take a look at the characteristics of each of them. So we'll start with carbohydrates. And if you're thinking, nom nom, I love carbs, yes, we are talking about those carbohydrates. Some of them are delicious. Monosaccharides are the simplest carbohydrates. Now, you might have heard of monosaccharides, it's a type of sugar. And when we are referring to carbohydrates, quite often we are referring to some type of sugar. So sugar monomers, each individual piece, are monosaccharides, such as those found in fructose, glucose, and honey. Monosaccharides can be hooked together by those dehydration reactions that we just talked about to form more complex sugars and polysaccharides. So again, think back, mono, like our monomers, that prefix mono means one. Poly means many. So just like monomer versus polymer, we have monosaccharides, which is basically one sugar, and our polysaccharides, which are the longer chains of sugars. The carbon skeletons of monosaccharides vary. Glucose and fructose are 6 carbons long, whereas other types of monosaccharides can have anywhere between 3 and 7 carbon atoms in their carbon skeleton. Monosaccharides are the main fuels for cellular work. You probably know that sugar is very important to your body. It's used as the raw material to manufacture other organic molecules. You can take a look here at glucose and fructose, 2 types of sugar, 2 types of carbohydrates. Notice that they are very, very similar, but they're going to be isomers of each other. So they're going to have the C and the O bonded in different places. And since the structure is different, the function of these two are going to be different. Many monosaccharides form the rings that we were looking at earlier. And remember that the ring diagram can be abbreviated. Whenever there's a point, we assume there's a carbon there at the corner of the ring. They can also be drawn with different thicknesses for the bonds. Just one line means single bond, 2 lines means double bond. And we can also shade in, notice here on the abbreviated structure, it's a little bit darker. If we make it a little darker, that means that there's more bonds. So you can see here, we can draw them in all different types of ways. So just be familiar with the fact that you're looking at the same thing. Scientists just write so many things that they like to be efficient. And so quite often we'll come up with some kind of way to abbreviate things to make their lives easier. So just be aware, we're showing you all the same thing in all three diagrams, so you can try to understand the differences there. Two monosaccharides are linked to form a disaccharide. The prefix di means two. And then polysaccharides are long chains of sugar units. So monosaccharide means one piece. Disaccharides have two pieces. Polysaccharides have many pieces, and the number can vary. Polysaccharides are macromolecules. They're very big macromolecules. They're polymers that can be composed of thousands of monosaccharides. So when we're talking about macromolecules here, we mean it. They can be very, very big. Polysaccharides can function as storage molecules. They can also function as structural compounds. They can affect the shape. Polysaccharides are usually hydrophilic. They're water-loving. I put towels here because what towels are made out of are compounds that are hydrophilic, so they attract water. Starch is a polysaccharide. It's composed of a glucose monomer and used by plants for energy storage. Glycogen is another type of carbohydrate. It's a polysaccharide composed of glucose monomers, and glycogen is used by animals for energy storage. Cellulose is another type of carb. It's a polymer of glucose, and cellulose is important. Remember this one. It forms plant cell walls. We're going to talk about plant cell walls quite a bit in future lectures, so remember that plant cell walls are made of cellulose. Chitin is not just an animal. Chitin is a polysaccharide used by insects and crustaceans to build an exoskeleton. There actually is an intertidal organism called a chitin, and it's called chitin because its exoskeleton is made of chitin. But chitin itself is the building blocks that insects and crustaceans use to build their exoskeleton. It's also found in the cell walls of fungi. All right, so that was a brief overview of carbohydrates. So now let's take a look at another type of organic compound called lipids. When we're talking about lipids, we're mostly talking about fats. So fats are a type of lipid, and these lipids are mostly energy storage molecules. So lipids are insoluble in water. They're hydrophobic or water-fearing compounds. They're important in long-term energy storage. They contain twice as much energy as a polysaccharide, and they consist mainly of carbon and hydrogen atoms that are linked by nonpolar covalent bonds. So they are nonpolar. The electrons are distributed fairly equally, which makes them hydrophobic or water-fearing. Lipids differ from the carbs that we just talked about and from the proteins and nucleic acids that we're going to talk about because they're actually not that big and they're not built from monomers. So when we were talking about carbohydrates, carbohydrates had a bunch of little pieces that were linked together to form the carbohydrate. Lipids are a little bit different. Now we're going to talk about three types of lipids: fats, phospholipids, and steroids. So fats are lipids that are mostly energy storage molecules. And a fat is a large lipid that's made up of glycerol and fatty acids. A fatty acid can link to glycerol by a dehydration reaction. A fat contains one glycerol linked to 3 fatty acids. We're going to look at a picture of this. And fats are often called triglycerides. The prefix tri means 3 because of those 3 fatty acid tails. So here's your glycerol at the top. You'll notice the orange box. That is glycerol. The bottom is one fatty acid chain, and they're linked together by taking a hydrogen from glycerol and the OH from the top of the fatty acid chain, doing a dehydration reaction, removing a water molecule, and then they link together. we're going to do this for each hydrogen at the bottom of glycerol. So here's one chain. What the lipid actually looks like is this. So it'll perform a dehydration reaction 3 times and attach 3 fatty acid chains. So this is the basic structure of a fat. Glycerol at the top, the 3 fatty acid chains at the bottom. This makes up our saturated fats and our unsaturated fats. And some fatty acids contain 1 or more double bonds, which is going to be the difference between saturated fats and unsaturated fats. So if a fatty acid contains 1 or more double bonds, it makes an unsaturated fatty acid. These have 1 fewer hydrogen atoms on each carbon of the double bond. And these double bonds can cause kinks or bends in the carbon chain. And remember, when you change the structure, you change the function. So this actually prevents them from packing together tightly and solidifying at room temperature. It prevents them from doing that. So our unsaturated fatty acids will typically be more of a liquid or an oil. Fats with the maximum number of hydrogens are called saturated fatty acids. Unsaturated fats are referred to as oils. Most animal fats are saturated fats. Our hydrogenated vegetable oils are unsaturated fats that have been converted to saturated fats by adding in hydrogen. This hydrogenation can create trans fats, which are associated with health risks. So you might have heard of saturated versus unsaturated fats in your food because this can actually cause an impact on your health depending on which types of fats you are eating. All right, so there's a brief overview of our fatty acids. So let's move on now to a different type of lipid called a phospholipid. And phospholipids and also steroids, that third type of lipid, are important lipids with a variety of functions. Phospholipids are very important because they're the major components of cell membranes. And we're going to talk about the importance of cell membranes quite a bit in future lectures, but it's what protects the cell from the outside environment. So we're going to have these, what we call selectively permeable membranes that only let some things through the membrane. And that's what's going to protect cells, is this cell membrane. And the cell membranes are made up mostly of phospholipids. Phospholipids are structurally similar to fats, where fats contain those 3 fatty acid tails attached to a glycerol. Phospholipids contain 2 fatty acid tails attached to a glycerol. So if you take a look at the image here, each blue piece is the glycerol. That's the phospholipid head, we refer to as a head. And then if you take a look, you can see it better at the bottom here. You'll notice that unlike fats that had those 3 fatty acid tails, our phospholipids here have 2. So you can actually see there's 2 phospholipid tails attached to each of the glycerol heads. So again, the blue parts are the heads, the yellow parts are the tails, blue parts represent the glycerol, the yellow parts represent the fatty acid tails. And this structure is going to be important when we talk about our cell walls, where there's one row facing this way and there's one row facing this way. So here is our phospholipid. Again, we've got our glycerol at the top. We've got our 2 fatty acid chains at the bottom. And actually here in phospholipids, we also have a phosphate group that's going to be bonded at the top here. What's important to note here is that the heads, so remember these blue pieces at the top and the blue pieces at the bottom, the heads are actually hydrophilic. They're water loving. The tails are hydrophobic. They're water fearing. This is going to be important when we're talking about the function of cell walls, of those cell membranes. So they're going to form what's called a phospholipid bilayer. Again, bi means 2, so there's 2 layers of phospholipids. If you take a look at this picture on the right, you'll notice the hydrophilic heads hydrophilic, water-loving, are going to be facing out on both the top and the bottom layer. And then the hydrophobic tails, those fatty acid tails that are water-fearing, are going to be in the middle, because surrounding both sides of this membrane is going to be water. So it makes sense that our hydrophilic, our water-loving heads, are going to be facing the water, and the hydrophobic or water-fearing tails are going to be safely in the middle where much water is not getting in. And it's just important to note that when you see something like this, where you've got a circle and then two little squiggles, that's our symbol for a phospholipid. So here it is again. We've got this whole part at the top, the glycerol bonded to the phosphate group, that is our hydrophilic head. And then we have our hydrophilic tails. Notice the kink down here. This is another way to look at it. So you can get very detailed with these diagrams here, where we have the glycerol bonded to a phosphate. We have our fatty acid chains down at the bottom. Here's a more simple way of looking at it, where we just have our polar hydrophilic head and our non-polar hydrophobic tails. Remember that polar molecules are usually hydrophilic, non-polar molecules are usually hydrophobic. Here is yet another way of looking at it. If you haven't caught on yet, these phospholipid bilayers are super, super important. So you're going to see it a lot. So here are hydrophilic heads. Again, they are polar. There's a slight positive charge on the top, a slight negative charge on the inside. We have our hydrophobic fatty acid tails in the middle, hiding from the water, and another layer of hydrophilic heads. Again, positive is on the outside, negative charge is on the inside. Now, our third type of lipid are steroids. Steroids are lipids in which the carbon skeleton contains 4 fused rings. So 4 rings of carbon that are fused to one another. Cholesterol is a type of steroid. It's a common component in animal cell membranes. It's the starting material for making steroids, including sex hormones. And that is your brief overview of lipids. The main takeaway from lipids, the most important parts are going to be the structure of both fats, where we had the glycerol bonded to 3 fatty acid tails, and especially those phospholipids, specifically the phospholipid bilayer. The phospholipid bilayer is going to come up quite a bit. So take some time either going back through this recording or looking through your book or watching the videos in the resources. Make sure you really understand what a phospholipid bilayer is. What is teacher talking about when she's referring to polar hydrophilic heads and non-polar hydrophobic tails? Why are they in the shape that they are, where there's the 2 layers of heads on the outside and the 2 layers of tails on the inside? Why is that? Why is it important? Because we are going to rely on that knowledge quite a bit in future lectures when we're talking about cell membranes. All right, let's move on to proteins. Proteins, again, delicious. Proteins have a wide range of functions and structures. You can probably name a bunch of proteins right now because they're very common in a lot of our foods. Proteins are involved in nearly every dynamic function in your body. We don't just eat proteins. They're very diverse. There's tens of thousands of different types of proteins, and each of those types of proteins has a specific structure and function within your body. You don't need to memorize all the pictures in the background. This is just to show you that there's a lot of different proteins. Proteins are composed of different arrangements of a common set of just 20 amino acid molecules. So in the background of this are our different amino acid monomers. And in future lectures, we're actually going to be learning how we take these 20 different amino acid monomers and link them together to form proteins. Again, you don't need to memorize everything that's in the background. If I'm asking you questions about it, I will give you a picture of the 20 amino acid monomers. But I want to show you that these are building blocks. So these amino acid monomers that you see in the background, there's only 20 of them. In all of nature, we've only found 20 of them, but you can link those 20 together in a variety of different ways. And depending on which order you put them together in, it makes different proteins. So all proteins are made up of those 20 building blocks called amino acid monomers. Now, probably the most important role for proteins is as enzymes. Enzymes are proteins that serve as catalysts, and they regulate virtually all chemical reactions within a cell. Now, the basic idea here, did I take that off? Yeah, perfect. Okay, so there's something that we call the energy of activation, which is basically the energy that it takes for a chemical reaction to get started. Because chemical reactions typically require energy to run. And it takes a certain amount of energy just to get started. Think about in the morning, you're laying in bed and you don't want to get out of bed, and it seems to take so much energy just to get out of bed. Then once you're out of bed, everything else doesn't require quite as much energy as that initial step of just getting out of bed. The getting out of bed is kind of the same as our energy of activation, where it takes a certain amount of energy just to get the reaction started. Sometimes that energy of activation is just too high and the reaction can't run on its own. So what an enzyme does is it lowers that energy of activation. So an enzyme functions based on its structure. So different structures create different enzymes. And the enzyme is going to go bond to a substrate. And basically what it does is, you'll notice this dashed line is the original energy of activation. So notice how much energy it takes to even get this chemical reaction started. It's a lot of energy. It might even be too much, so the reaction might not go. An enzyme is going to come in and it's going to lower the energy of activation. So now, instead of having to get energy way up here at the top, all this energy, the enzyme is going to bring it down to the solid line, so it doesn't take as much energy to get the reaction started because of the enzyme. So enzymes lower the energy of activation. They lower the amount of energy required to start a reaction, making it easier for reactions to go. Then once a reaction starts, just like when you get out of bed, it doesn't require as much energy to keep going. The good thing about enzymes is they come in, they do their job, and then they leave. Enzymes are not used up in a chemical reaction. They do not become part of the chemical reaction. All they do is come in, lower the energy of activation, and then get out of there. And since they're not actually involved in the chemical reaction and they're not used up in the chemical reaction, enzymes are then available to go do this again and again and again. This is actually super important. I don't know how long I've been talking here, but I'll take a moment to tell you one of my favorite stories. I used to work at the San Diego Zoo, and koalas, they look super cute. They're actually kind of mean. I've met some koalas. They're kind of grumpy. But I've actually been surprised at how many of my friends, a lot of my friends are not, they're not called keepers anymore. They're now called animal care specialists, FYI. But a lot of my friends are animal care specialists at the zoo, and I'm surprised by how many have actually turned down koala keeper positions, because koalas eat eucalyptus and only eucalyptus, right? Just eucalyptus, that's all they eat. Well, there's over 100 different types of eucalyptus. If you look outside, you'll probably see eucalyptus growing. So when the zoo first got koalas back in the 1920s, the keepers were like, well, sweet, there's eucalyptus everywhere. I'm gonna bring it in, feed it to the koalas, and have free food for the koalas, right? So they fed it to the koalas, and this wasn't just a problem at the San Diego Zoo. This was actually a problem at zoos all over the world. The koalas either refused to eat or they died. And that is because koalas on their own cannot digest eucalyptus. That energy of activation to break down what's in eucalyptus is too high, and the koalas can't do it on their own. So they have this special relationship with these organisms in their gut, with these that have enzymes. They release enzymes that help to lower the energy of activation and help them to break down the compounds found in eucalyptus. Well, I mentioned that there are different types of enzymes. Each enzyme can only catalyze one type of reaction. So each enzyme can only do one specific thing. And for koalas, what this means is that each particular type of enzyme that they have can only break down one type of eucalyptus. And so they have these little bacteria in their guts that release these enzymes, and each type of bacteria only releases the enzyme for one type of eucalyptus. The only way these koalas get these bacteria, because they're not born with them, is once they're born, mom releases a special type of poop that has those bacteria in it. Little baby koalas go and eat that poop. They then get that bacteria in their gut. The bacteria starts releasing those enzymes, and the enzyme helps them break down their eucalyptus. Typically, koalas only have a handful, like 5 or 6 types of bacteria that they pass on to their kids. And since those enzymes are specific, they can only eat the type of eucalyptus that's associated with the bacteria and the enzymes they have in their gut. So even though there's over 100 different types of eucalyptus, each koala can only eat a handful of those types. And if it's the wrong type, they either know they can't eat it, so they don't eat it, or they eat it, they don't have the enzymes for it, so they can't break it down. And it's like basically then eating nothing, and then the koala can die. So the koalas at the zoo don't all eat the same types of eucalyptus. So those keepers have to be able to identify all the different types of eucalyptus and know which koala can eat which types of eucalyptus. So koalas are actually super hard to care for because of this. And so that is what I hear is why a lot of the keepers will actually prefer to work with other animals because of enzymes. Think about that, enzymes. So there you go. Okay, back to proteins. So there are other types of proteins, including transport proteins, defensive proteins, signal proteins, receptor proteins, contractile proteins, structural proteins, and storage proteins. For the most part, they do pretty much exactly what they sound like. Transport proteins transport things. Defensive proteins, like antibodies, defend against things. Signal proteins, like hormones, send messages. Structural proteins, like collagen, help with structure. Storage proteins store things. So read through this. I'm not going to spend much time on it because they basically do what they say they do. So just be aware there are all of these different types of proteins. They are very important to your body. So just understand a little bit about what they do and the fact that they exist. Now the function, we mentioned the word function a lot because it is important. The function of different types of proteins depend on their individual shapes. Structure affects function. The shapes of proteins are super important. A polypeptide chain contains hundreds or even thousands of amino acids linked together by what we call peptide bonds. So just remember that proteins are made up of amino acids linked together by a certain type of bond called a peptide bond. You don't need to get too in depth with what a peptide bond is. Just know that that's how amino acids are linked together to form proteins. And the amino acid sequence, remember there are those 20 amino acids that are linked together by peptide bonds to form all the different types of proteins. That sequence makes the polypeptide assume a particular shape. And again, shape affects function. So the shape of a protein is going to be very important. There are actually 4 different levels of protein shape that we're going to touch on briefly. Again, I'm not terribly worried that you understand everything about each of the 4 levels of shapes. Just be aware that there are 4 levels of shapes of a protein and know basically what each level is. But I don't expect you to get too much more in depth than that. The main takeaway here is that shape affects function. All right, just make sure you get that out of this lecture, that shape affects function. So the shape of a protein is going to change its function. And since proteins perform so many functions, we are going to talk a little bit about protein shape. So this is one of the computer drawings that you'll see a lot for proteins. They're usually very messy. The purple represents the polypeptide, the chain of amino acids that form a protein. And you'll notice they do all kinds of crazy things. They do swirls and they overlap with each other and they get all tangled up. They can form these temporary bonds you see here. We refer to those as grooves. You're looking at the same thing here, just a different type of drawing. This is a little more detailed where you can actually see all the different atoms bonded together in the molecules of the protein. Again, here's the groove. So you can see that they're kind of the same shape here. We're showing you the same thing. So just be aware there's different types of computer models depending on how much information you want. And since shape affects function, if a protein's shape is altered, it can no longer function. All right, I want you to understand this more than the difference between these two. Be aware there's different ways of drawing these. That's what I want you to get out of this part. What's more important, and that I want you to understand, is that if a protein shape is altered, it no longer functions. Shape affects function. You change the shape, you change the function. You change the shape too much, it can no longer function. One of the ways of changing the shape of a protein is by heating it. Think about how we have to heat meat before we eat it. This is one of the reasons. And you'll notice on the left, here's proteins are just all over the place. So here's a drawing of what the protein is supposed to look like. When you heat it up, you change its shape. So you actually can break some of those bonds. We call it denaturing. And when you start breaking those bonds, you change the shape, which makes it so the protein does not function anymore. So in the process of denaturing a protein, it unravels, it loses its shape, and it loses its function. Proteins can be denatured by changes in salt concentration, by changes in pH, or by high heat. So again, proteins are made from those 20 amino acids that are linked together by peptide bonds. So here's an amino acid. So we're gonna talk about amino acids briefly here. Amino acids are made up of a chemical group, which we symbolize by R. So take a look at this yellow dot. This is the R group. And amino acids are basically going to be an amino group. Remember, we talked about those functional groups and how they're important and they're going to keep coming up. Get your cheat sheet out. So we have an amino group and we have a carboxyl group. They're going to be bonded to an R group. Amino acids are basically going to be an amino group, a carboxyl group, and then some kind of R group. And what that R group is going to determine which amino acid this is. So all amino acids are going to have an amino group and a carboxyl group. The difference between amino acids is the R group. So the R group is what we want to pay the most attention to because it's that R group that's going to determine which amino acid it is. And again, there are 20 of them that are used to make up proteins. So amino acids are classified as either hydrophobic, like leucine here, or hydrophilic, like serine and aspartic acid. Again, remember, hydrophobic means water-fearing. Hydrophilic means water-loving. Amino acid monomers are linked together through those dehydration reactions that are going to join the carboxyl group of one amino acid to the amino group of the next amino acid. This creates those peptide bonds that I mentioned earlier. Additional amino acids can be added by the same process to create a chain of amino acids that we call a polypeptide. So let's take a look at that. So we have one amino acid and we have another amino acid. Notice that they are fairly similar. The difference is going to be in whatever this R group is. And we're going to take the carboxyl group of one amino acid and the amino group of the other. We're going to perform a dehydration reaction. So we're going to take the OH group from, or out of the carboxyl group of one amino acid. We're going to take the hydrogen from the amino group of the other. H2O. So we're going to remove a water, and that's going to make a peptide bond. All right, so you take out a water molecule, that's going to link the two amino acids together by a peptide bond. And again, it's the OH from the carboxyl group of one amino acid and the hydrogen from the amino group of the other amino acid that get removed as a water molecule to form that peptide bond, linking the two amino acids together. So let's take a look at how a protein's functional shape results from 4 levels of structure. So I mentioned that there's 4 levels of structure. I got you all excited about it. Here they are, our 4 levels of structure. Again, I'm not terribly worried that you know every detail about every level of the structure of a protein. Because let's be honest, how many of us are actually going to be talking about the quaternary structure of a protein in our everyday lives or even in our professional lives? But the fact that proteins are so important to your body and that there are so many proteins and that the function of a protein is because of its shape, it's important to have a basic understanding of the 4 levels of shape because it's just so important to how our body works. So here we have our primary structure, which is the different amino acids that are linked together by these peptide bonds. So the primary structure is just going to be which different amino acids are linked together. That's our primary structure. Our secondary structure is going to be how that chain is basically shaped. So how they're linked together by hydrogen bonds. So secondary structures are maintained by hydrogen bonds between atoms of the backbone. So they can form these spirals, they can form pleats. So our primary structure is just which amino acids are linked together in which order. Our secondary structure is either going to be an alpha helix if it's in that spiral shape, or a beta pleated sheet if it's in that pleated sheet. That's our secondary structure. Our tertiary structure, that third structure, is how the R groups, remember amino acids differ by their R groups. Tertiary structure, or our third level of structure, is determined by how those R groups interact with each other. And our quaternary structure is how the polypeptides are associated into a functional protein. So this is as deep as I want you to get into this. There are 4 levels of structure of a protein. This is important because of the structure of a protein determines its function. If you change the structure, you affect the function or make it not function at all. Primary structure, that first structure, which amino acids are linked together in which order. Secondary structure, our second structure, is either, is it an alpha helix, that's spiral, or is it a beta pleated sheet? Right? How these different pieces are interacting together through hydrogen bonds. Tertiary structure, our third structure, the interaction between R groups. Quaternary structure, our fourth structure, how the polypeptides are associated into a functioning protein. There's proteins. All right, here is our last group, nucleic acids. So let's take a look at nucleic acids. Notice the word nucleic refers to the nucleus, and one of the most important roles of the nucleus is to protect DNA, deoxyribonucleic acid. Just start to soak that up, deoxyribonucleic acid. We're going to talk a lot about it. By the end of this course, you are going to be very familiar with DNA. So for right now, just prepare yourselves for DNA and RNA. But there are 2 types of nucleic acids: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. They're very similar to each other. You will be expected to be able to name the difference. So we're going to go over that. But to introduce you to the concept, DNA and RNA are both nucleic acids. You've probably heard of DNA because it determines what your cells do. You know, it tells a liver cell to be a liver cell, a tongue cell to be a tongue cell. And the primary difference between DNA and RNA is that DNA is made up of a sugar called deoxyribose. RNA is made up of a sugar called ribose. There are some other differences that you will be expected to know in the future, but as we're introducing it, just know the main difference between DNA and RNA is the type of sugar. So the amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as a gene. Again, this is just a brief introduction to nucleic acids. We are going to get up close and personal with genes in future lectures. So just for this lecture, I don't necessarily need you to understand everything about a gene. I just want you to start getting familiar with the terms. So that's your goal for this. Because nucleic acids are fairly detailed, and we're going to get into those details, the point of this lecture right now is for you to just start understanding the basics and getting familiar with the terms. The more you hear them, the easier it will be for you to understand them. So the amino acid sequence of the polypeptide is programmed by a discrete unit of inheritance known as a gene. So a discrete unit of inheritance, it's going to be a piece of DNA that is being passed on to your kids. That's known as a gene. Genes consist of DNA, deoxyribonucleic acid, which is one of our types of nucleic acids. DNA is inherited from an organism's parents. So when you have kids, you are passing your DNA on to your offspring. DNA provides directions for its own replication. DNA has instructions on how to make more DNA. And DNA programs a cell's activities. It tells a cell what to do. You're going to be a liver cell, you're going to be a tongue cell, you're going to be an eye cell, by directing the synthesis of proteins. So DNA basically tells the cell which proteins to make. DNA is the set of instructions for how to build everything in your body. DNA does not build proteins directly. We do not go from DNA to protein. There's something in between. And that something in between, which we call an intermediary because it's in between, is RNA, ribonucleic acid. DNA is transcribed into a type of RNA in the nucleus, and then RNA is translated into proteins in the cytoplasm. We are going to talk about transcription and translation in detail in future lectures. For right now, here's what I want you to get out of this. DNA does not build proteins directly. Rather, DNA is transcribed. Remember that word. DNA is transcribed into RNA, and also remember that this transcription happens in the nucleus. DNA is found in a cell's nucleus, and if the cell is functioning properly, you will not find DNA outside of the nucleus. So DNA is transcribed into RNA in the nucleus. That specific type of RNA then leaves the nucleus, it goes into the cytoplasm, and is translated into proteins. DNA is transcribed into RNA, RNA is translated into proteins. That's the part I want you to get right now. We will talk about how this happens in future lectures. All right, so here's a gene, which is basically a part of DNA. So we have a particular sequence of DNA that is a gene. Remember, genes are what is passed on to our kids to tell their cells what to do. That DNA is transcribed into RNA. Notice DNA is usually a double helix. So there's 2 strands of DNA. RNA is usually single-stranded. So we are going to transcribe 1 strand of DNA into RNA. It's going to leave the nucleus, and it is then going to be translated into proteins. And it's going to do that 1 piece at a time. 1 amino acid linked together at a time to form a protein. Nucleic acids are polymers of nucleotides. So nucleotides are the building blocks of nucleic acids. You're going to be putting many nucleotides together, that's the polymer, to form nucleic acids. So DNA and RNA, both composed of monomers, those individual building blocks called nucleotides. Notice here, DNA and RNA are actually fairly similar. They both have a phosphate, they have a sugar, and they have a base. The phosphate group is going to be the same. Remember our phosphate groups from earlier in the lecture. So the phosphate group of both DNA and RNA is going to be the same. The sugar is going to be different. They both have a sugar, but DNA has a type of sugar called deoxyribose, whereas RNA has a type of sugar known as ribose. And then they each have a base. We're going to take a look at that coming up here. So there are 4 bases of DNA: adenine, which we shorten to the letter A; guanine, which we shorten to the letter G; cytosine, which we shorten to the letter C; and thymine, which we shorten to the letter T. So those are the bases of DNA. RNA also has 4 bases. It has adenine, guanine, and cytosine, just like DNA does, but in RNA, instead of thymine, we have a base called uracil, and uracil is shortened as the letter U. So this is the basic shape of our nucleic acids. We are going to have our sugar bonded to our phosphate, bonded to a sugar, bonded to a phosphate, bonded to a sugar, bonded to a phosphate, making a sugar phosphate backbone. And then sticking out from that backbone are the nucleotides, those bases that we just talked about. This structure is important because it affects the function. That the backbone is made up of alternating sugars and phosphates, and then the nucleotides stick out from that backbone. And I want you to take a look. Is this DNA or RNA? How can you tell? We'll take a look here. It has T, it has that thymine. Remember down here, thymine is only found in DNA. If it was RNA, it would be uracil. So that's one of the easiest ways, and teachers love to ask you that question. Are you looking at DNA or RNA? The easiest way to look is if you see thymine, the letter T, it's DNA. If you see U for uracil, it's RNA. In DNA, which is typically double-stranded, you have 2 strands of that sugar phosphate backbone. So here's one, then you can see the other. And they're going to be bonded together through those base pairs. Notice here in the middle that A is bonded to T. They do form pairs. Adenine always bonds to thymine. So A always bonds to T. I remember it by the word at because A always bonds to T. And then cytosine always bonds to guanine. So C always bonds to G. This is important because another thing the professors like to do is give you a long list of letters like A, T, T, A, C, G, 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 C, T, A. Now write the complementary strand. And you can, because if you see an A, you know that the other strand of DNA, the complementary strand, if that is an A, the other one is a T. If one strand is a C, the complementary strand is a G. So you can actually write this out. If it was RNA, RNA is typically single-stranded, but you're still going to be using those base pairs in order to make more RNA. And we're also going to be using that when we are talking about translation, when we're going from RNA to proteins. And so in RNA, C and G still go together, but A in RNA would go with uracil. So adenine would go with uracil, because remember that in RNA, we have uracil instead of thymine. So the most important part here is the sugar phosphate backbone is going to be on the outside. Sticking out on the inside are going to be those base pairs, and they are going to pair up adenine to thymine, A to T in DNA, or adenine to uracil in RNA, and then cytosine to guanine in both DNA and RNA. But remember, the DNA stays double-stranded. In RNA, you're only going to be seeing this if you're making more RNA or you're making proteins. Whew. All right, so now you should be able to describe the importance of carbon to life's molecular diversity. You should be able to describe the 6 chemical groups, those 5 functional groups, and the methyl group. Explain how a cell can make a variety of large molecules, which we call macromolecules, from a small set of molecules. Define monosaccharide, disaccharide, and polysaccharide. Remember, mono means 1, di means 2, poly means many, and explain their functions. Define what lipids are, what phospholipids are, especially the phospholipid bilayer, hydrophilic heads, hydrophobic tails, know it, love it, and steroids, and explain what their functions are. Explain how trans fats are formed in food, know the basic difference between a trans fat and the other types of fats. Describe the chemical structure of proteins and the importance of proteins to cells. Remember, structure affects function. Describe the chemical structure of nucleic acids and briefly explain how they relate to inheritance. At this point, I want you to know that, remember those different base pairs, right? That those letters, which order the letters are in, a discrete section of that is a gene that is passed on to your offspring. The order of the letters tells that offspring what to do. Be a liver cell, be a tongue cell, be an eye cell. That's what I mean. And then in your book, if you'd like to get into how lactose tolerance or intolerance has evolved in humans, there's a section on that in your book, but we did not cover in lecture because the other things are what I really want you to focus on. So that is the end of lecture 5. Thank you for listening. I'll see you soon.